eukaryotes. Very important. It works differently in prokaryotes. Eukaryotes. So generally, but not always, generally you're initiating from the first, the most 5' AUG on the message. Yeah, up there. Is there, say again? Yeah, the other AUGs in the protein are just going to come from the binding. So there's actually a different tRNA. It's not stuck to the initiator tRNA. It'll be stuck to the F1. And there, any AUG inside, downstream of that message is just going to come from the binding. Alright, so what does this say about the 5' untranslated region? Going to find an AUG in the 5' untranslated region? No. No. So you don't want to get mutations that lead to a new AUG upstream. If you do that, what can happen? You're going to initiate early. What's the consequence of initiating early? You get a longer protein, what's the worst consequence? Frame shift. Absolutely. Now it does happen that the first AUG in some eukaryotic transcripts is not the initiation site. There are transcripts in eukaryotes that can initiate from a couple of different AUGs. This process is a little bit leaky. In some cases, these other AUGs are in frame. And so you make basically the right protein with a little less amino acids and fiber. A little leaky. Questions about initiation? In eukaryotes, yeah. So, in eukaryotes, there are times when translation does not initiate the first AUG, initiate downstream a little bit. If that downstream AUG is in frame, you're still going to make a protein. It's going to have a few less amino acids on it. If it's out of frame, you're going to make a completely non-sensible protein and finally have a protein in this style. Alright? Yes? The extra amino acids completely change the protein structure and function. Not always. They could. If you add amino acids in part of a protein, let's say you added amino acids in part of a protein that was the active site for an enzyme, that could be critical. But actually in the lab, we stick amino acids, extra amino acids on proteins all the time. And it's amazing how much stuff you can stick on the end of a protein and still have it be functional. How would you do that? Thinking back to your protein lecture, if I wanted to stick a big set of amino acids at the end of a protein and have it still be functional, how would you do that? Yeah. Yeah, so if you're using protein domains and they're connected by these unstructured linkers, you can actually stick some pretty big chunks of protein onto the end of the protein together. And that flexible linker, it won't affect the structure of the other domains downstream. That's exactly right. The reason we do that often if we're interested in localizing a protein by fluorescence, we can stick what's called epitope tags under the end of the protein, or GFP, green fluorescent protein, is a protein you can add to the end of your protein, and it fluoresces, so you can see where your protein is. So the answer is, sometimes it can affect you, sometimes it doesn't. Other questions? All right. Let's talk about prokaryotes and operators. All right, so prokaryotes have what are called polycistronic messages. What that means is, and this is an old term that sometimes you see in the literature, This means that you have a single mRNA that's coding for a bunch of different proteins. And we should say unrelated proteins. Because a single immature RNA, as we talked about in eukaryotes, can code for several different proteins. How? Alternative splicing. This has nothing to do with splicing within prokaryotes. Alright? They don't splice. How does this happen? Well, the way you do this, if you have 3 different genes on the same mRNA, which is what you get out of an operon, remember the lac operon has 3 different genes on the mRNA, all controlled by one promoter. Very efficient way to regulate these things. What happens is, you've got to have internal ribosome binding sites. So if they initiate a translation just like you do in eukaryotes, could you make an operon work? They initiated the first AAV from the 5' could you make 3 separate proteins from the same RNA? Who says yes? Okay? We'll come back to you there. Who says no? So if you initiated the first 5' end of an mRNA that's got 3 different genes on it, you're only going to get 1 peptide. So in prokaryotes, they actually have a mechanism for initiating internally on the RNA. So what does that look like? In prokaryotes, this initiation complex is not recognizing 5' cap and moving down this RNA and finding the first AAV and then initiating it. If you do that, you make this protein, you get to the stop, and the whole ribosome will fall off. You couldn't make these other 2 proteins on that message. So the way initiation works in prokaryotes is that initiation complex, I'm sorry. So the question is, why can't you make it start the first AAV and go? What's going to happen? Well, I'm going to initiate the first AAV. I'm going to make protein alpha. And then what's going to happen when I get to the stop point? This falls off. Alright? The way initiation works in prokaryotes, we're not finding the 5' end and then scanning for those AAVs. They're actually looking for specific sequences upstream of the AAVs called the Shine-Dalgarno sequence. Shine-Dalgarno sequence actually binds to the small subunit of the RNA by complementary base pairing. Alright, so the sequence in the small subunit of the RNA that recognizes the special sequences upstream of the AAV. So initiation can occur here, here, and here, simultaneously, done by complementary base pairing. Now there are some sequences that are preferred around the initiation AUG in eukaryotes and help that ribosome decide whether or not it's going to initiate there. In this case, it's very, very specific. This is how an operon works. This is why you don't generally see operons in eukaryotes. Even in worms, we generally don't see operons in eukaryotes because the initiation and translation works differently. Nothing else. It has nothing to do with transcription. It has to do with translation. Questions? Yeah?
Eukaryotes and operons use the same mechanism, or do they use the
Yeah, so the question is, I set myself up for that question. Eukaryotes that do operons, do they utilize the same mechanism? They don't. And I'm not sure exactly that the organism, the eukaryotic organism that I know the best that has operons is worms. I don't know how initiation, translation initiation works in worms. Although there are cases in other eukaryotic cells where you can initiate not at the first AAV, but at internal AAV. Right? And you can use the same machinery. You can use the same machinery to do it. So how do you do that? If I wanted to initiate this sort of eukaryotic mRNA and I want to initiate a liver, how would I do it? What's the best way to initiate internally? knowing what you know about initiation in knowing what you know about initiation process, how do I initiate internally? What is the initiation complex recognizing? Let me raise your hand. Yeah. Go. What is the initiation complex recognizing? It's recognizing these initiation factors that bind to the 5' cap and the poly A tail. Alright? So, okay, if I want to initiate internally in an mRNA, how would I do it? Try it again. I want to initiate. You can't initiate internally on an mRNA. In a eukaryote. How would you do it? Knowing now how you normally initiate, how would you do it? You want to get either of these initiation factors binding internally in the mRNA. Yeah. Subsequent stop and start codons. Yeah, so that's an interesting idea. Yeah, you could have a start codon really close to the stop codon. Yeah, the rate termination, we haven't talked about termination. That's tough to make happen. But we'll talk about that in a second. Yeah, so yeah. What? Add the poly A tail in the middle, so you have to chop that mRNA. And then there you go. You got it. Then you mark the end of your message right where you want to start handling. Good call. But basically, you want to get those proteins, those initiation proteins, to stick somewhere else on the mRNA. Now, there are viruses that do this. The polio virus does this. And so we've already kind of gone through the thread. So the polio virus, these viral genomes that do this to try and trick the cell into recognizing its own thing. So here's the normal initiation complex. It's recognized by the 12 ribosomal subunit. They have sequences called internal ribosome entry sites, IRES sites. And these sites fold up in the RNA to a secondary structure that mimics 5' cap, interacts with poly A. There's the initiation factor. That initiation factor is tricked into recognizing the secondary structure of the RNA instead of 5' or instead of this region right here that interacts with the 5' cap. You trick the cell into doing this with an RNA structure, and then you can initiate right here with your own poly A. So you get full length RNA here. Internal ribosome entry sequence. You see it in viruses, you'll see it anywhere else. All the viruses. So we actually take advantage of this in the lab. We can put these IRES sequences onto it. We can make these polysystronic messages. Helps us watch. We use the other proteins as process and proteins are interested in. So it does happen. We use the old machinery, but you have to trick it into binding these proteins that normally bind at 5' cap and poly A. Yeah. Yeah, this is normal initiation. In this case, you still have this initiation factor 4G that's recognizing poly A binding proteins, and now you're getting it to stick to this wacky RNA structure instead of this 5' cap that has been initiated. What ribosome binding sites? We're talking about eukaryotes. Are they the same in prokaryotes? So are these Shine-Dogar sequences the same? Yeah. Oh, there's going to be a consensus. There's going to be an efficient, there's ways to regulate efficiency by how close you match the consensus in the Shine-Dogar. It's how close do I match complementary base pairing-wise the sequences in the small subunit of the ribosome. That's what's recognizing these initiation signs. Can you vary from those and still initiate? Yeah. The farther you vary, the less efficient initiation is going to be. Other questions? Yeah. Recognize Shinedown garments. There you go. The ribosome, small cell unit of the ribosome, the initiator of thymine, and its special component. We're all scanning, looking for these ribosome binding sites. Yeah. So prokaryotic translation is so much faster because the elongation is 20 amino acids per second
That doesn't make any sense.
per ribosome. Right? Here you're just putting how many ribosomes are you going to use to initiate on this one right there. So it's on a per ribosome. So the answer is no, because elongation is faster because yeah. They all use the same stop codons. There's 3 different stop codons. I mean, there's going to be a stop codon somewhere here, there's going to be one here, there's going to be one there. There's 3 different proteins. Yeah. Yeah, so ribosomes, the question is, does one ribosome create each of these 3 proteins in order? Probably not. Well, it should make more sense when we get to termination, because at termination, everything releases from mRNA. Now, whether that same ribosome then reassembles those, possible, but not. Yeah. The alpha protein overlap to the beta protein? Yeah, the answer is yes, it can. So realize, given this mechanism for initiating translation, E. coli can make itself a much more compact genome by having all of these proteins coded on a single RNA with a single promoter and operator. It's a very, very compact and efficient way to evolve your genome. Why have humans done this? Maybe they don't need to right there. Great question. I assume you have an answer to that? Right, so the question is given how much faster eukaryotic or prokaryotic translation is versus prokaryotic translation, what's the fidelity? I don't know. I tried. I can do that on my own, just about not. Perhaps look that up for me. There we go. Yeah. Ribosomes that bind to the protein. So ribosomes binding to the mRNA. So what's, I don't understand your question. The ribosomes are all the same. It's not the same one doing this one, this one, and this one, but there's not a bunch of different kinds of small subunits. Same kind of site, same kind of small subunit for the ribosome. Yeah. It's not the same ribosome from there. Yeah, it's a little different. Yes.
Is there like transcription where you can have several ribosomes transmitting at the same time? You have one start and then later you have another one start while the other one's still transmitting?
Yeah, there's no rules as far as I know for when these ribosomes can hop onto this transcript. So they're probably hopping on and translating and hopping off at different times. Other questions? termination. It is. Alright, let's talk about termination. Release factor. This is what recognizing stop codons. What type of molecule, so it's recognizing stop codon, what type of molecule would you expect release factor to be? What's the most obvious call? Yeah, you're right. I would say TR. We've already established mine. So, I mean, the easiest way to do this for me would be to have a tRNA that didn't have anything stuck to it. Now, recognizing the stop codon, it turns out, you're right, the protein guys go ahead. They're just so release factor is indeed a protein, and when you get to a stop codon, it's a protein that's recognizing, in this case, the UAG. So you get to UAG there, and all these tRNAs are going in, and nothing binds to it, and nothing's being added because nothing's complementary. Release factor goes in, recognizes that stop codon. As soon as you get recognition of that stop codon, you get termination. So that first thing that's going to happen is that join polypeptide chain is going to that bond is going to get broken, and the polypeptide chain is going to get released from that last tRNA. And then that large ribosomal subunit is going to shift over, and the whole thing comes apart. So as soon as you hit that stop codon, you release the polypeptide chain from the tRNA, and then that large subunit shifts over, and everything comes apart. That's probably why you're not going one after another in the program. Yeah. Question is, how come the protein doesn't have to be attached to some kind of nucleotide sequence in order to recognize the codon? Can you think of another case in which a protein is recognizing a specific set of nucleotides? Can proteins recognize specific nucleotide sequences? What's one example? Restriction enzymes would be one. Right? They recognize specific sequences of nucleotides. Got it? What's another one? Yeah. Transcription factors also recognize very specific nucleotide sequences. So proteins can recognize specific nucleotide sequences on their own without the help of another nucleotide chain. How do they do that? So the question is, how would proteins recognize specific nucleotide sequences? Are they different? Is the chemistry of each of these bases different? Yeah, so you have a protein that recognizes the chemistry of those bases, either forming a good bond with the proper set of bases, or a weak bond without the proper set of bases. Same way, all the same rules in which proteins recognize each other, they can recognize parties or deities. Yeah. So why does it, so the question is, why does the release factor go on the A side and not the P side? Yes. Why doesn't it all fall apart there? Because you want to make sure that you trigger the release of that polypeptide chain that's going to the telomere. So part of what happens is protein is facilitating the release of that bond. You can't make a peptide bond, so you can't make the peptide bond and then cleave. There's no peptide bond here to make. So it's cleaving that. Once you cleave that, it's a matter of structural changes in the ribosome that help it come from. So biology doesn't always do it in its logical or efficient way. It does it in a way that works. And you want to make sure there's time to get that protein released from that tRNA. Otherwise, you're going to have a tRNA hanging off your protein. Yes? Are there different kinds of proteins that recognize different stop sequences? I don't know the exact answer to that. I would guess there are probably three different release factors that recognize the different stop codons. It could be one that recognizes all three. I don't know. Do you have a question?
No, there are a couple different, I think there are 2 different release factors, and one of them recognizes 2 of the stop codons. If I remember.
Oh, one, yeah.
Yeah, there's some weird.
So Chris says there's 2 and one of them recognizes 2 different stop codons. Yeah, real quick.
So, I guess the book it says that prokaryotes, there's 2 and they both recognize overlapping receptors. Alright. That's termination. So, getting back to this whole idea, one of the common themes in the course has been structure function. This is a tRNA right here. Remember, I said for me, it made no sense that the release factor was going to be a specialized tRNA. This is the structure of the protein called release factor. We talked a lot about RNAs having enzymatic activity because they can adopt secondary structures that mimic what proteins can do. Here's a case in which a protein has adopted a structure that mimics a tRNA. So it can fit into those sites. So it goes both ways. Talked a lot about RNAs forming secondary structures. Here we have a protein that mimics that RNA structure. Over RNA. Right, cool. Alright. So the last thing I want to show you is that as with transcription, you can have multiple, on the DNA during transcription, you can have multiple RNA polymerases transcribing the same gene at the same time, making multiple transcripts. On many highly efficiently translated mRNAs, you can get what's called a polysome. And this is an mRNA with multiple ribosomes in various stages of translating that single mRNA. So each of these is making a polypeptide. So from a single mRNA, you can get multiple proteins. And these RNAs are translated with different efficiencies. That can be affected by the sequences. Remember, we talked about sequences in the 5' UTR that affect the efficiency of translation. That makes a lot of sense, right? Because that 5', the very 3' end of the 5' UTR is the initiator AUG. So having sequences around there that help that ribosome bind will make it more efficient. Having sequences around there, especially secondary structures that prevent that ribosome from binding, are going to inhibit initiation. Any single mRNA can get multiple proteins made from it. There are a lot of RNAs in cells that you find in 1 to 5 copies maybe per cell. You can still make a fair amount of protein. Questions? Yeah. It happens in both. This in particular is what? Can you tell me? Yeah, you can tell by the RNA. Yeah,
The fragment bind to the polyethylene cap as soon as the mRNA leaves the nucleus.
so you mean the initiation factors?
Yeah.
As far as I know, the initiation factors are in the cytoplasm, but I don't know that they're not actually binding before export.
No, you're right after as soon as it leaves.
Yeah, I can't tell you that it's my understanding that initiation factors are found in the cytoplasm, so binding after export. But they actually be binding before export in some cases. I don't know the answer. All right, we'll see you guys on Wednesday.